The overall goal of this procedure is to activate single somatosensory neurons in larval zebrafish. This is accomplished by first preparing an optic cable and connecting it to a DPSS blue laser. The second step is to inject the transgene into 1 to 2 cell stage embryos. Next, screen for embryos expressing ChEF-tdTomato in somatosensory neurons and mount them in agarose. The final step is to deliver a short pulse of blue light for single neuron activation and to record the resulting behavior.
The main advantage of this technique over existing methods using channelrhodopsin 2 is that this approach can be used to elicit behavioral responses from single neurons activated up to at least 4 days post-fertilization. Though this method is being used to provide insight into the function of specific somatosensory neurons in larval zebrafish, this approach could also be adapted to study behaviors elicited by any zebrafish neuronal population, such as interneurons or motor neurons. This optogenetic approach could be used to activate specific neurons in vivo, characterize functional development, assess the effects of mutations or pharmacological blockades of specific proteins on behavior, and map downstream neuronal networks leading to distinct behavioral responses.
To begin this procedure, create a storage unit for the optic cable by melting the tapered neck of a glass Pasteur pipette over a Bunsen burner to make a 150-degree angle. Next, using a wire cutter or razor blade, carefully cut the optic cable into 2 pieces. Each piece should have one exposed end and one end with an FC-PC adapter. Then, strip the optic cable down to the cladding by removing the fibre jacket